Hello and welcome to today's class of Introduction to Mechanobiology. So, in the last class we had started discussing about the nucleus and its structure, and majorly I introduced this complex called link complex, which stands for linker of nucleoskeleton and cytoskeleton. Okay. So, this link complex just make sure that if this is my nucleus, okay, you have a physical connection from actin or microtubule or intermediate filament cytoskeletons to the inside of the nucleus through various connections. Okay. So, if I zoom in, so the nucleus actually is a double membrane system. You have outer nuclear membrane and inner nuclear membrane. So, inside the inner nuclear membrane you have the network of Lamin proteins. These include Lamin A and B, okay. And outside, so to the cytoskeleton you have these links which are mediated by nesprins. So, this establishes a direct physical connection from outside the nucleus to the inside of the nucleus. Okay. We also spent considerable amount of time in discussing how expression of lamins. So, when we say lamins, you have two major classes of lamins, lamin A's, but it is encoded by the same gene, but because of splicing you have two forms, lamin A and lamin C. Okay. So, this is collectively written as lamin AC, and you have the lamin B form found as B1 and B2. Okay. So, what has been recently demonstrated is that cells which reside in soft tissues contain more of lamin B, while cells which reside in stiff tissues, they contain more of lamin AC. Okay. So, if you plot the stoichiometry of lamin A to B, okay, so you get this relationship where if x axis is tissue elasticity, okay, and y axis is A to B stoichiometry. So, you have this scaling relationship where below 1 here in soft tissues like brain, okay, soft tissues like brain, you have cell types like glioma cells, they would reside here where lamin A to B ratio is less than 1, while stiff tissues like cartilage, bone, muscle cells and even mesenchymal stem cells contain high ratios of A to B. Now, mechanically A and B operate at different entities. Okay. Lamin B operates like a spring, okay, and lamin A operates like a dashpot or a viscous object. Okay. So, collectively it is expressed as this, where this is your B response and this is the A response. Okay. So, your short term, whenever you deform the nucleus, okay, when you deform the nucleus, you have a short term response which is lamin B dominated and a long term response which is lamin A dominated. Today, so this was the first part of what I discussed in nucleus. The second part was the importance of A in dictating cell motility or cell migration under confinement. Okay. So, since high levels of A lead to stiffer nucleus, so high levels of E actually impede migration. By migration I mean confined migration. Okay. But if you have A too low, then you might have damage to the nucleus. Okay. So, in the next two lectures, including today, I will discuss two things, that how the nuclear deformations eventually influence gene expression patterns. okay, and this will be mediated by chromatin dynamics. Okay. So, I have already taken an example of the YAPTAZ transcription factor, which translocate to the nucleus. In response to mechanical signals. So, in the context of atherosclerosis, so this is oscillatory shear stress. Okay, in response to oscillatory shear stress, YAPTAZ was shown to translocate to the nucleus. There are other such transcription factors. Notable among them means NF kappa B signaling, STAT signaling, okay, and MR TF signaling pathways. This also exhibit this trait where they do translocate to the nucleus. And what do these translocation induce? Okay. So, these actually lead to alterations in gene expression. Okay. So, I would like to discuss two papers today briefly. Okay. One paper discussed how does cell shape influence gene expression. So, you are both aware of the earlier paper when we were discussing about mesenchymal stem cell differentiation. We showed that when cells are plated on small patterns, then they tend to differentiate into adipocytes, okay. And then on big islands they tend to become osteoblasts. Okay. So, this is cell shape mediated. So, what the authors did in this particular study was they took a variety of shapes of microfabricated islands coated with fibronectin. So, these are fibronectin islands, okay, and they generated various geometries, okay, different shapes, triangles, rectangles and circles, and they also altered the sizes of these. Okay. So, there were two things that they varied: the cell shape and cell size. So, cell shape was changed, modified by changing the shape of the islands that they used, and cell size was controlled by the dimensions of the islands. Okay. And then they asked that what happens when we plate. So, they used NIH 3T3 fibroblasts. Okay. So, they asked how do signaling alter when cells are cultured on these different patterns. Okay. So, first they did a comparison between circular and elongated morphologies, okay. And what they found was, in this case of circular patterns, so circular is a more isotropic geometry versus this is a more polarized geometry. Okay. This is non-polarized or isotropic. Okay. So, what they found that on these circular patterns they had upregulation in, okay, cell division proteins, apoptosis genes and regulators, and negative regulation of cell matrix addition. Okay. Again, so there were some other signals which are down regulated on the circles. So, when I say up regulated, this is up regulated with respect to the elongated geometry. Okay. Similarly, there were several things which were down regulated. You had actin cytoskeletal machinery, all the actin cross linking proteins were down regulated. Proteins involved in cell migration, their expression was down regulated. cell substrate addition was perturbed, okay, so on and so forth. Similarly, they observed differences. So, they used the triangular geometry. Okay. They compared a small triangle with a big triangle, okay. And what they found was in the smaller triangles there were several signaling pathways which were upregulated. Okay. These include regulation of transcription, RNA metabolic processes, cell motility, and adherence junctions. Okay. So, now all these differential changes in gene expression that were observed with increase in sizes. So, there was a correlation. So, the differential changes in gene expression with increase in cell size were largely mediated by the MRTFA SRF pathway. Okay. So, SRF is serum response factor. So, when you know that when we culture cells, we always supplement with serum. Okay. Serum has multiple factors including growth factors as well as ECM proteins which benefit cell cell growth. Okay. So, SRF pathway is involved in all of these processes. What they then checked was whether there was any alteration in histone acetylation, okay, H3 acetylation levels at lysine 9. Okay. So, this is typically written as ACH3K9, H3K9 is a histone acetylation. So, what they observed was when the shape was different, so long as the area was same, okay. So, the nuclear volume and H3K9 acetylation was identical. Okay. So, even though the shapes were different, nuclear volume and acetylation levels were identical. However, for the same shape, when they increased the size, okay, so they found increase in nuclear volume increased and H3K9 acetylation also increased. Okay. So, these experiments actually revealed that across the conditions there is a very linear relationship between H3K9 acetylation levels and nuclear volume. So, based on this, they suggested a pathway in which any cell geometry that you regulate will influence the actin cytoskeleton, okay. And by actin cytoskeleton, when it is perturbed, what it will change is contractility, and this contractility will eventually act upon, okay, HDAC3 localization, cytoplasmic localization and nuclear shuttling MRTFA. Okay. all of these eventually perturb chromatin condensation levels. Which dictates the gene expression patterns. Okay. So, this paper was published in PNAS 2013. They have a more recent, they have followed up on those experiments, okay, and then they chose these two geometries, okay, again a polarized geometry and a circular geometry. Okay. So, this they wrote as large polarized geometry or constrained isotropic. Okay. So, this is constrained isotropic, okay, and this is large polarized. So, through this they wanted to ask that how does cell geometry influence nuclear deformability and chromatin dynamics. Okay, so, this was the question. So, what they did once again was they plated cells on these islands. They plated 3T3 cells, fibroblasts on these islands. So, you have two geometries, elongated and circular. Okay. On what, so, what they observed was on this geometry they show prominent stress fibers, indicative of increased contractility, while this was much less prominent stress fibers in this case. Okay. And what they observed was on these two, if you were to compare the nuclear geometry, okay, here if I look in side view, the nucleus will have a geometry like this. Okay. So, in other words, it is elongated, okay, and its height is less. Here for the rounded geometry, you would assume that the nucleus would also be rounded, okay, and its height will be more. Okay. So, this is less, this is more. Okay. So, given this, they next asked that, okay, so this is static shape. Okay. How does the nuclear dynamics change? Okay. So, when what they did by for understanding nuclear dynamics, okay, what they did was they transfected these cells with GFP H2B. Okay. So, H2B is a histone protein which is known to bind to the DNA. Okay. So, as a result, so when you transfect cells with H2B, you can detect the entire nucleus, round or elongated. It will give you signal throughout the nucleus. Okay. And then once they transfected these cells, they can actually track the outline of the nucleus. Using thresholding. Okay. And they asked that how do these shapes change as a function of time. Okay. So, as a consequence, you can have, if this was a geometry at one given age, at one given time instant, you can track. So, let us say this top surface is the area and this vertical axis is my time. Okay. You would get a 3D surface, okay, where every section that you take will give you a given geometry. Okay. So, what they found was on the LP islands, okay, large polarized islands, the nuclei, this surface that they generated, this was very smooth. Okay. The surface was very smooth. In contrast, on the circular islands, okay, the nucleus, if I were to draw the surface, if you do the chymograph, you get a surface which is much more ragged, okay, suggesting that nuclear area change, okay, nuclear area change is much more pronounced on the constrained isotropic islands. Okay. So, when I talk about nuclear area, it is actually the projected nuclear area. Okay. So, to quantitatively detect how much does the nuclear area change, what they did was they plotted the percentage fluctuation in the area, in the projected area. Okay. And so, this is your time axis. So, compared to on these islands, okay, on the LP islands, if this was the fluctuation, on the circular islands the fluctuation was much more. So, this is on the constrained CI islands and this was on the LP islands. So, this confirms that the area fluctuation is significantly higher on the rounded islands. Okay. So, this shows, so the higher amount of fluctuation, okay, so the higher amount of fluctuation is linked, they linked it with nuclear deformability. The idea being, if something is rigid, then it will retain its shape for extended periods of time, while if something is floppy, particularly within the cell, there is always so much of activity going on, the nuclear will be subjected to lot more changes in area as a function of time. Okay. What they then asked was, what is driving these fluctuations? Okay. So, they did experiments where they perturbed actin in two ways, either they destabilized the effectin network with cytokinesin D, or they stabilized, okay, with jasplakinolide. So, what they found, okay, so what they found is this. So, my y axis is percentage area fluctuation. Okay. So, compared to the rectangle, so if rectangle, this is your area of fluctuation. Okay. On the rectangle, when they added with cytokinesin D, okay, when they added cytokinesin D plus cyto D, this fluctuation increased. Okay. But on the circular patterns where fluctuation was significantly higher, when they added cytokinesin D, okay, this decreased. Okay. So, this suggests that these effects of polymerizing drugs are highly non-monotonic. Okay. They also did experiments. Okay. So, they did not did experiments with Lamin AC knockdown cells. They also found that with this, on the circular patterns, the extent of area fluctuation increased. Okay. Increased. They did experiments by perturbing Nesprin, okay, and still they observed area fluctuations. Okay. So, based on this, they concluded that perturbations, okay, of the physical links partially affect the amplitude of fluctuations, because even when they perturbed with actin, it dropped, but it did not go, or it increased, but it did not go. Okay. So, as the last step, what they did was they tracked chromatin dynamics by labeling with H2B JP and tracking foci. Okay. They tracked the heterochromatin foci within the nuclei, and what they found, okay, was that these chymographs, okay, so this heterochromatin foci exhibited faster dynamics in CI compared to LP. Okay. And this was sensitive to actomyosin contractility. Okay. One more thing that they found was within the same nucleus. Okay. If you have these multiple foci, they found that the motion of these foci were actually correlated. Okay. The motion of the foci were correlated. Okay. And these foci, though they the extent of correlation was perturbed when you treated them with drugs like actane or blebistatin, but when you remove, then again, okay, when you remove, these actions were removed. Okay. So, the foci trajectories, okay, the foci movement was spatially correlated. Okay, and sensitive to cyto D. Okay, but after washout, okay, the positions and the correlation map is re-established. So, what all these experiments suggest is there is an imprint. So, there is a structure of chromatin map. Okay. So, there is a structure of chromatin map, okay. And what they find, okay, that this structure, okay, is controlled by multiple forces like contractility. So, you have actin cytoskeleton, Lamin AC, nesprins or link domain proteins, and so on and so forth. Okay. So, these collectively regulate, okay, the telomeres, okay, which serve as the endpoints of the chromatin. Okay. With that, I stop here. I would suggest to read two papers as reading assignment. Okay. So, both these papers make use of a geometrical pattern and then study how you have these epigenetic modifications happening as a function of shape and size, and how telomere dynamics and within the nucleus how dynamics change when you perturb these cytoskeletal elements. Thank you for your attention.